In the same way that barcodes on your groceries help stores know what's in your cart, DNA barcodes help scientists attach genetic labels to biological species to identify them from one another. Yes, you heard it. Even organisms have barcodes, DNA barcodes. In the past, scientists had to rigorously study different species under a microscope and attempt to distinguish them by their physical features. However, this method proved to not always work, because two different species can look extremely similar to one another, making it hard for scientists to identify them by their morphological characteristics. Yet, it is the analysis of their DNA sequences that aids scientists in distinguishing and grouping different species. DNA barcoding is a method that uses a short genetic marker in an organism's DNA to identify at the species level. In fact, one of the professors from the School of Life Sciences and Lee Dak-sum Yip-Yu-shin R&D Center for Chinese Medicine in our university, Professor Shaw, has been researching on the authentication of Chinese herbal materials by DNA barcoding. Depending on the type of organism you are working with, scientists have come to the consensus of using particular regions that are effective at discriminating samples to the species level. These regions are known as barcoding regions. For plant-based herbal materials, internal transcribed spacer 1, ITS1, internal transcribed spacer 2, ITS2, in the nucleus or chloroplast PSBA tRNH intergenic region are the DNA barcodes. Take the Asian ginseng, Ginseng radix, and the American ginseng Panacea quinquefoil radix as an example. As they look quite similar to one another, DNA barcoding using the ITS region is applied to differentiate them. Now let's say you have been given an unknown ginseng sample and you have no idea whether it is the Asian ginseng or the American ginseng. How do you go from this unknown sample to an identified species? Well, DNA barcoding has 6 main steps: DNA extraction, polymerase chain reaction PCR, agarose gel electrophoresis, DNA sequencing, sequence alignment, and species identification. Firstly, DNA extraction. DNA extraction is the process of isolating DNA from other cellular components such as mRNA and proteins. This is a key step because without high-quality DNA, PCR amplification will not be optimal. PCR is a technique used to amplify small segments of DNA such as the barcode region, ITS, using primers. With repeated cycles of PCR, millions of copies of the original barcode region can be generated. Thirdly, agarose gel electrophoresis. To verify the success of amplification of our barcode regions, PCR products are loaded onto an agarose gel immersed in a buffer. Under an electric field, any amplicons and residual primers will migrate towards the positive electrode and separate according to their molecular sizes. With a dye that binds with DNA and a DNA lather, which is a mixture of DNA fragments of known sizes, we can verify if our barcode region ITS was amplified with a band at the expected size, which would be around 600 base pairs. Once verified, the concerned DNA band will be sent for DNA sequencing. DNA sequencing is the process of determining the order of nucleotide bases, ATGC, in a piece of DNA. Now that we have the DNA sequences of our barcode region, we are ready to analyze our sample by sequence alignment. To do that, we have to head over to NCBI's BLAST, Basic Local Alignment Search Tool. BLAST is a computer algorithm that identifies similarities between biological sequences by comparing nucleotide or protein sequences to a database of sequences. By inputting the nucleotide sequences of our sample obtained from DNA sequencing, we can find sequences that BLAST identified to be similar to our query sequence, and then identify our sample by the best hit. In this case, our unknown sample would be the American ginseng Panacea quinquefoil radix. DNA barcoding is an effective tool for species identification and offers several advantages such as only needing small amounts of sample, not affected by age and growth conditions of samples, and has high resolution power. Hopefully, in the future, DNA barcoding can not only increase the pace of species discovery, but also further facilitate taxonomists in classifying different species.